of second day international conference. Now it's my distinct honor to invite Dr. M. Sudhakar sir, our revered principal and professor at Malla College of Pharmacy, to grace the dais and take his rightful place among us. I request Ms. Srikala to present a bouquet of flowers to honor our respected principal, Dr. Sudhakar sir. Your gesture symbolizes our deep appreciation for his unwavering dedication and leadership in fostering excellence within our institution. May I have the honor to call upon the stage our distinguished speaker, Dr. V. Venkatesarulu sir, Managing Director, Neuheit Technologies Private Limited, former President at Dr. Reddy Laboratories. A symbolic welcome with a flower bouquet would be greatly appreciated. Next, it is my privilege to invite our speaker of today's session, Dr. Ramaligeswara sir, Senior Consultant, Vidhu Pharma Solutions, former Head Operations, Novartis. May I request Ms. Kirtana to extend a warm welcome to Dr. Ramaligeswara sir with a flower bouquet. Now I kindly request Dr. M. Sudhakar sir to speak few words on this occasion.
So good afternoon to all my dear students. Are you energetic? Yes. Hope you had a good lunch. And lunch means not only food. Whatever you had in the today's morning session, all brainstorming sessions by Dr. Chandraya and Dr. Jagan Mohan. Excellent lectures we had in the today's morning. And another giant here, pharma expert, pharma eminent person, Dr. V. Venkatesarulu, who was the professor in the Kakatiya University and presently is the MD for Neuheit Technologies Private Limited and former president of the Dr. Reddy's Laboratories. Enjoy the session. He is the author of one important book, as you all know that. Which book? Biopharmaceutical book, I think. Right, sir? Biopharmaceutical pharmacokinetics book. Excellent book. It's all followed by many students. Okay, sir? I welcome you, sir. Sir.
Good afternoon, sir. I would like to introduce our guest, Dr. V. Venkatesarulu garu. Sir is a postdoc from US and faculty member for 16 years in Kakatiya University and guided around 12 PhD students and several MPharm students. He has about 80 international and national publications and 15 patents to his credit. He is the author of a book entitled Fundamentals of Biopharmaceutics and Pharmacokinetics. He is a dedicated researcher and co-founder of Neuheit Pharmacotechnologies in 2013 and 17, which is engaged in development of complex generic and differentiated products from ideation to commercialization. He developed platform technology in Microspheres project, selection design with DOE and QPD principles. He established bio-relevant dissolution methods for control release and complex solid orals. He is an expert in bio-waiver approach. He is a consultant to many companies across the world in clinical and bioequivalent studies. I welcome you, sir, and please.
Guru Brahma, Guru Vishnu, Guru Devo, Maheshwara, Guru Sakshat, Parabrahma, Tasmai, Sri Guravim. Good afternoon. Good afternoon. Thank you. So I have been given a very big challenge of not making you to sleep. After a heavy lunch. Really a big challenge for a speaker who takes a talk after lunch. But I will try my best to see that you will be active, enjoy the talk. But there's only one humble request. If you fell in sleep, sleep quietly, don't snore, don't disturb others' sleep. Okay, thank you. Now the topic today I have taken for the discussion is the pharma industry academia interaction, that is collaboration. Now since, am I audible? Since the topic is the future of pharmacy, innovation, collaboration and transformation. So you have to innovate, collaborate and transform is the interest if you want to really go into the future of the pharmacy. Good? But where we stand on pharma industry academia collaboration in India is an important point we need to discuss. First of all, is there any use to have pharma industry academia interaction? Yes or no? How many are for yes? No. There are few people they say no. So you believe that there is a need for the collaboration between the industry and the academia? Yes, then let us see how it is working in other countries and why it is not working in India. Then how we should transform ourselves to achieve this goal. That is the whole subject of today's discussion. Because the idea is that unless the industry academia comes together, there is always a problem of innovation that we are going to see now. May I have the next slide, please? Well, this is where you can see that industry academia collaboration requirement is identified by our people, even it is discussed and debated in the parliament, and they have come up with a plans and budget to encourage the industry academia interaction. That means people know there is a need, people know that there are provisions, but still it is not happening. That is the problem. The next one is, there are certain funding agencies from the government. If we go together, industry and academia, if they come together and propose a project to the government funding agency, they are ready to give the fund. But how many are being utilized by us is a question. The third point is, if you look into the global scenario, in US or advanced countries, many drugs, novel drug delivery systems are introduced by big pharma only with good collaboration with academics, many. In a way, there is always a synergy when industry and academia comes together. Today we should know why this synergy is great, how it is coming, and how to use this collaboration for the benefit of the Indian people for having innovative and affordable medicines. The next one point is that now why India is failing when every country is able to make good industry academia interaction, collaboration and bringing very affordable and innovative medicines. There are three bullets, you can identify that. Lack of mutual trust and lack of strengths and weakness of each parties. See, whenever it is a collaboration, it's just like wife and husband, isn't it? Two people have to come together and live together. Similarly, industry scientists and academic scientists should come together, work on the same project. When it is possible, when you trust each other, isn't it? And also you know what is your strength and what is the complementary skill or the strength other person has. So your weakness can be taken care by your counterpart and their weakness can be taken by you, so that it becomes a perfect collaboration. But both of them don't know what is the strength of other party. That is the problem. And I am fortunate in the sense I worked in academics for 16 years and also worked in a good company like Dr. Reddy's for 8 years. So I could see now an academic research on my own and also industrial research on my own, luckily. So therefore, when I am there in academics, I was not greatly appreciating the industrial research. And when I came to industry, then I understood that what is the academic interest, strength of the academic scientist. So I will take one very important answer given by the great Dr. C. Narayan Reddy. You know C. Narayan Reddy? Hello? He's a good lyric writer. You know that part. But actually he was a professor in Usmania University. Isn't it? So now somebody asked him a question. You are writing books and poetry as a professor also. Now you are writing lyrics in a mass Telugu movie. Now the question is, which is easy? Or this academic writing or industry writing? Film industry writing. So because he worked in both the areas. The answer was very, very, very apt and good. That will help us to understand. What he said is, I don't know how many of you are thorough in Telugu, understanding of the Telugu area of literature. It always contains a grammar, well-defined grammar. If I have to say in Telugu, then Vyakaranam, Samasalu, Sandulu, Chandassu, Yati, so many things are there. Now, when somebody want to write a poetry, great poetry in Telugu, in academics, he has to go to the higher level of thinking and understanding and writing. That means you have a freedom of subject, whatever you want to write, but it has to be the highly competitive in the peers review. This is one part. Coming to the second part. There is no Chandassu, there is no restriction on the language. You can use English, Hindi, Telugu together. You can write the way you want. But what is the but? By the time you are going there to write a lyric, already tune is completed. That means the music composition is already over. That means whatever you write, now it should fit into the music. Am I right or wrong? Hello? So that means one constraint is that you can write, but they have to put into the music notes. Immediately second point comes. What is the second point? This situation is already fixed. The hero, the heroine, what is happening there, the situation is fixed. Whether it is a village or it is a town or it is a club, situation is fixed. Now he has to write a lyric that fits into the situation. The third one is image of the hero. Chiranjeevi or a big hero, you cannot write just like that. You have to keep his image into the system. Or if you are writing to Brahmananda, you have to think different. So he says, you tell me I have constraints here, music is composed already, the situation is defined, the hero, heroine are fixed, now I have to write a lyric that suits all the three. Therefore, there is nothing like easy research. Research is always a challenging area. You have both the challenges, freedom in industry, challenges, freedom in the academics. Now we need to dig out what are the freedoms available in academics, what are the freedoms available in industry. If they are complementing each other, better to combine together so that you can do wonders by collaboration. That is the basic point. Coming to the second one, divergent goals. Divergent goals means an academic scientist wants something else. An industry scientist wants something else. Your goals are different. How can you collaborate? I want to go to the south, you want to go to the north. We cannot collaborate, isn't it? So these are the important points you need to understand. What are the different goals they have and how to bring a common goal for both of us. Next one is lack of encouragement. Natural. This is everywhere. Am I right? Hello? Encouragement is only the missing element. Somebody was telling, common sense is most uncommon. Hello? common sense is most uncommon. So therefore, now coming to the encouragement is the most important element that is missing either from the management or from the government. So that unless there is encouragement, support by these people, you cannot. These are the three important things that are lagging the India to be back in the position for the industry academy interaction. Therefore, the miracles are not happening because of that. The next one, please. Wow, advantages with academia. You can see that now the academicians should be proud of these advantages. Many skilled resources. At one place of Malla Reddy College of Pharmacy, I can find a chemist, I can find a pharmaceutical person, I can get a pharmacology. Everybody is available here. If I have a problem to be discussed, having different subjects requirement, all are here. You won't find like this in industry. They have to be dedicated. So therefore, multi-skilled, not only that, the PhD students, the M.Pharm students, and multi-skilled people will be available, and the resources are very experienced faculty members, 10 years, 15 years, teaching experience, research experience, and that to several people, 30, 40 people sitting there. So therefore, there is a multi-skilled, experienced faculty sitting there. Then, they always look for the advantages in the science and technology. So what is new? Journal reading, publications, they publish their findings, they read the new articles, understand what technology is coming out into the science. So this is called in line with the progress. Now always they look for the innovative thinking to get the solutions to the problem. You might have seen in the talk when Jagan Mohan was giving, can I combine two absorption pathways into one product so that I will get a benefit out of it? So nobody is going to question you why you are working in academics, because you can hypothesize. Always academics are looking into a different way of solving the problems so that the innovation is very high. The next one. There are also some weakness or lacks. You can look into it. Lack of time management. Sorry to say, generally academics chalega. Okay, if not today, tomorrow. That is, time management is not so serious in the research. So therefore, that is the most important problem in the academics. I am talking about India, please note that. And lack of intellectual property knowledge, meaning you can do anything for academic research, you can enjoy the research, but that you cannot do for the industry, because somebody might be having his knowledge patented in the form of patent, and you cannot use patented knowledge to create some product and go and sell. But academicians are not worried about this. This is most important. But when I want to collaborate with an academician, what I have to look in is any research what we are doing, any product we are developing, it should not go into anybody's patent. Otherwise you will be sued and then dragged into the court. But that knowledge is lacking in the academics. Similarly, the regulatory knowledge. What is the meaning of regulatory knowledge? Is to get approval for a tablet formulation or a capsule formulation of a product. What are all 1, 2, 3, 4, 5 required to get the approval? They don't look into that. They only try to solve the problem of bioavailability enhancement. Oh, no, that is not going to help. Along with that, you have to show that the materials you are used to increase the bioavailability are safe. They are also safe. They are non-toxic. You have to study that, but they don't study that because only I want to improve the bioavailability. But when I want to go to the market, that won't work. So regulatory knowledge is missing in the academics. Next one is industrial exposure is not there. What is the problem? See, in the lab, you may prepare 500 tablets, 1000 tablets. The same science won't work when you are producing 1 million tablets. So the academician cannot visualize that unless he works and understands how very big batches of tablets are manufactured. Then when you are developing your process in the lab, you think about the problems you are going to get in a manufacturing facility, then you design your process accordingly. That is missing in the academics. And improper maintenance of the labs. The labs are very poorly maintained. There is no good laboratory practices in the labs in academics. A GLP requirement is there, good laboratory practices. Every day calibrating the equipment, calibrating the things, and then doing it, maintaining, preventive maintenance. So that is not possible, because in academics you do today one experiment, it will come next year. Am I right or wrong? In second year, the one experiment is conducted. They only again next year, second year only they do that experiment if that equipment is used. So there is no regular maintenance. And last, as usual, I said encouragement. Management is not going to encourage any researcher do the research. Teacher has to go to the class. Have you completed the class? Syllabus is completed, exams conducted, grades are given, blah blah. But no research requirement is required. And that is the reason where the teacher is not motivated to involve himself in the research. And are you giving, instead of 16 hours, 2-3 hours leverage to go and do the research? The answer is no. So there is no encouragement. That is one point I want to emphasize. The next one, please. Now let us come to the advantage of the industry. What are all the industrial advantages you can see here? They have the IP knowledge, that is intellectual property, regulatory management. They do have most important thing. Any research point we are talking about, the academician is not aware whether this product I am developing is useful to the market, will it generate any commercial value? But industry people perfectly know that if you crack this, if you bring this product, it will add value and you will get the value. They maintain good laboratory practices, good manufacturing practices that are suitable for the regulatory environment. And they know perfectly how to scale up and make a commercial batches, millions of tablets, day out, day in, with high quality. And they know best project management. I will take one example from the corporate behavior, how they do the best management. I was vice president in Dr. Reddy's for some time. There are other senior vice presidents, all those things. Do you know? For all others, they give the freedom to go and have a cup of tea. They can just go and dispense and have. But for vice presidents, a fellow will bring the coffee to the table. Are you following me? Hello? Are you here? We have an agreement that you should not sleep, okay? So therefore, what is happening here is, we feel that proudly somebody is bringing coffee, we need not to go there. That is not the corporate mentality. The salaries are very high for the vice president and senior vice president. They don't want to waste 15 minutes. This fellow going and having cup of tea and coming back. So that is not the provision given as a respect. Remember, provision given as a best corporate practice, because they count every point. So a high salary person should not waste 15 minutes time for taking tea. So therefore somebody brings and puts on your table. So that is the project management thinking of the corporates. The next one, please. Now there are concerns with industry also. They are not perfect. They do have problems. What is that? Number one is always the routine way of doing the things. But that lacks the innovation. Innovation is gone. So that means think differently, out of box thinking, blah blah. They won't happen there. Focus on quick business potential. Everything is, if I give a project to some academician, can you give in 6 months? Can you give in 4 months? The bargaining is on a delivery point, not looking into the depth of the problem, timelines, etc. Because the thing is, they always look rupee in, rupee out. So how much I am putting, how much it will come back. Limited resources. They don't want to have too many people, and everybody has to be engaged to 120%. Are you following me? 120%, not 100%. So that is the reason they take the limited resources and finally timelines. If you ask all my industry friends, they know that officially you can go exactly as per the time, okay, 9 to 6. It never happens. It will be 7:30, 8, sometime 10, you don't know. So here they want the same tight timelines in the academicians, that is going to be a problem. So this is now what I discussed is advantages and concern with the academics, advantages and concern with the industries. In addition, and two different people have to come together, and when it is possible, when the two people fell in love, hello? When there are certain common things between them, otherwise it never happens. No? There are certain common things both like, then it leads to a pairing. Otherwise pairing not happens. So similarly, if industry scientists and the academic scientists want to come together and work on a collaborative project, there should be some common things that are happening. So at this at this common point, I get a very important joke I heard. One of the colleague was telling, we always fight, myself, my wife, always fight. Then the fellow was telling, at least I think we have 2 times compromised because they have 2 kids. So the next one. Now the goal divergence means you have a one goal, I have a different goal, then we cannot do. You can see in academics, the most interesting thing for the academicians is, what is that? Innovation. I want to do differently. I want to do something different because I want to publish and show my knowledge. When it goes to the publication, the peers are going to be very, very watchful. What is that new thing you are added in your paper? That is important for publication. So it is nothing but innovation. But whereas our industry friend is looking for what competitive edge I am getting over other fellow to get more money from my product. It's called a competitive edge. The second one is to be at cutting edge. So the academic scientists, when they are working seriously, they want to see that science is great. For example, I will tell you one very interesting proposal what we got. See, 80% success was happened in the product. They want to go for the 90%. So they are innovated in such a way that the cost gone to 4 times. So they look into the innovation only. The business fellow will look for 10% improvement, you want 4 times cost. So 80 rupees will become 240 rupees. Will somebody is going to afford for your 10% delta? No, sir, with 10 milligram I am struggling to get 90%, so 80% bioavailability is there. I said, what to do that? Put 11.5 milligram, it will come to what is required. So I can afford to put 1 milligram more, which is cheaper than innovating to improve the 10%. But he want to show that this cutting edge technology improves the bioavailability. So that is the conflict we have. This is right, but that is not going to add value. So if we come together, your knowledge of improving to 80%, my knowledge of commercial thinking of putting 1 milligram more, making the things happen, will give the good product. Then now, the reason how to go to market quickly is my thought. You want to have a cutting edge, then both will be conflict. The next one, please. Well, difference in design objectives. One fellow, everybody has their own objectives, and let us see what is the difference here. Once, if I take scope, for academician it is open-ended. You know, meaning of, with latitude, the meaning of latitude is the distance between the north to south pole. So that means you have everything in your hand, you can think slowly, you can do whenever you want. It's called the leverage. You have every leverage, open-ended, but a very tightly defined target scope will be there for the industry. So there is a conflict. One want to be very tight, one want to be too liberal. Similarly, time management, as I already told, is highly flexible academicians, but for industry people, it is a very strict time management. And resource management, multitasking. In academics, what happens is, multitasking means, for example, you are involving your PhD student, now you have to go for the PhD work, then he has to do the extra work of his project, and he has to prepare for his pre-PhD exam. And so multiple tasking, or one fellow will come to do the part of work and the other fellow will come do the other part of work. So that is called the shared type of thing. But where in industry they dedicate, means it is their deliverable, they have to do one or two, they not. And cost control, in the cost control, academician is always looking for what? Subordinate to goal attainment. What is the goal attainment? Publication, grant approval, project writing. But here it is a budgeted approach. They fix the amount, in this amount, how to make it happen. There is a difference between the goals. The next one. And even their performance is measured differently. If you look in, peer recognition is the most important aspect for the academicians. What is the peer? Peer means the one who is working equal to you. Equal to you is called the peer. So, when somebody was giving, I will take one important dimension of the psychology and the emotional management of our own thing, because when the peer has come, remember always we encounter with only three people. In our life, professionally, we encounter with only three people. One is above you, one is equal to you, another is below you. Do you think anything more than this? Hello? Is there any other possibility? No possibility. So then when you are handling these situations, encounters, respect always your seniors, discuss and debate with your peers, love your juniors or the small people. If you practice these, you always will be great. Okay, now coming to that they want the peer recognition. So the same professor, somebody in the cadre, they have to appreciate a very good paper and then they will be happy. But for this fellow, industry, no, market share, penetration to the market, getting more market share. The sustained support for research and graduates is only simple happiness for the professor. That I should get research grants, my research scholar should get their fellowship. For the industry fellow is very quickly I need to go to the market and grab the market. So these are the performance differences that we are looking at. So they are all inbuilt into the system. Unless we come together, have a common agenda, we are never going to succeed. The next one, please. So conflict, what is the meaning of conflict? Conflict means between you and me there is always a difference of opinion, is called the conflict. Here the conflict is confidentiality. When the research work is going on in academics, we don't know guarantee that it will be very carefully preserved without leakage to other areas, because once the invention is leaked, then you are gone. And the second one is the indemnity. So if any loss is incurred because of the mishandling by the academician, the company will be at loss and nobody is there to protect it. That is called indemnity. These are the two problems when industry want to enter into academics. When academic want to enter into the collaboration, they are very, very serious about the project scope, because you are asking increasing the bioavailability is okay, but you are asking me to do the safety assessment, you are saying the stability assessment. So, but the thing is, to go to the market, they are all required. But problem statement, we say that increase the bioavailability. But it doesn't mean that it ends there. Therefore, these academicians say that no, you said only increase bioavailability, now you are saying so many things. That always happens. And the project management, very tight schedule, Monday morning, 11 o'clock, you said you will give the report, what happened? This is the question from the industry. So that tightness will be there. This is again the conflict. The last one, next. Now, how to improve? Yeah, naturally, how to improve is definitely we have to understand each other. When wife and husband have to live together very nicely and happily, sacrifices, as well as same time some gains and losses have to be balanced. So now here, first of all, my proposal is that industry scientists invariably should come and teach in academics, do certain research activity with the academicians. With their own academic research is going on. For example, you are doing some nanotechnology activity, you are developing a nanotechnology on your own in academics, an industry fellow should come and involve in this project and also involve in teaching at least 1 hour per week or something for quite some time. Then he will understand what are the capabilities of the academics, advantage of the academics, and also the weakness of them. Now he will appreciate certain areas where it is not possible in industry. Similarly, vice versa. Academician, I have seen several people, those in formulation also, they don't know how is the RMG, rapid mixer granulator, how it operates. It's very painful because you have plenty of pharma industries here, pharma industries. You can at least go for a week or something, you can request and have a round and involve in certain activities. Then you get an industry perspective. Then you understand their strength also. So that is the second bullet. Management should encourage, and it's only an appeal. You cannot do anything, but management should think this collaboration is in a long way is going to benefit both of them very, very high, and Indian population at large. And most of the industries should try to give some funding to the academic small projects, slowly to make them to acquaint with the industrial research. If not big projects, small projects can be given. A core committee constituting of industry, academia, management of this and management of this. A core committee if you form, then these people are there, 2 from the academics or 2 from the industry, form a committee, and now they can coordinate any conflict management, difference of opinions, and finally everything should be to the best of our ability in black and white. What is the scope of the project, timelines, budget expected, how to share the profits, what should be given, everything can be written down. So therefore that is the way only that is possible. That means the management of academics, the management of industry should come together, have the first collaboration of exchange of the scientists for some time, and then slowly build trust, rapport, and then start working in joint projects. The last one. Next slide, please. Spirit de corps. What is the meaning of this is? The common spirit existing in the members of a group and inspiring enthusiasm, devotion, and strong regard for the honor of the group. Actually, the sentence always simplify. If I have to simplify, together we win. Either English translation is together we win. So always when we have togetherness, then only we will win. So therefore, to do that, there is always a need for feeling belonging. We are all one team. Like people might have seen Check the India. Anybody? Movie Check the India. Hello? At least 1 or 2 Check the India. Shah Rukh Khan. Yeah. So there, what is there? The spirit de corps. This is the spirit. That means team is above all, not the individuals. The pride comes when all of us do some sacrifices, go extra mile, work each other. So then only it happens. So therefore there is a long way, my dear friends, and I am very disappointed that we are not still able to come out of that shell. And so many drugs were introduced because of universities, because of academic research. There is a big list at the end after thanks. But even novel drug delivery systems, many are invented. And all are happened only in US and Europe and some advanced countries. And interestingly, when you go and look into them, there is an Indian name in the team. Indian name in the team. That is the most unfortunate thing. When Chandrasekhar got the Nobel Prize for black hole theory, he is from India only. But once he has taken the US citizenship, it goes to the US. Amartya Sen Nobel Prize goes to the UK. Got my point? Therefore, this is the problem. So let us come together, make something that is possible. Thank you.
About successful pharma innovation, academia collaboration. At this moment, it is my privilege to introduce our next speaker, Dr. Ramaligeswara sir, who held distinguished roles in the field of pharma industry for a span of 30 years. He is expertised in setting up R&D labs, pilot plants and manufacturing units. Dr. Ramaligeswara sir worked for reputed companies like Novartis, Dr. Reddy's, Natco Pharma, Eisen Drug Research Solutions, Channel Pharmaceuticals, Biological E. Notably, he received prestigious awards like Professor S. Rangaswamy Gold Medal, CSR Award for Engagement, Global Award for Simplification, Platinum Award GU Expansion U Project. I now invite Dr. Ramaligeswara sir to address the gathering.
Good evening all. I think I am the last night watchman. I think it's always tough. I think he has at least a better chance to more listeners, but I think I hopefully I will be able to make you interesting. At the outset, I would like to thank the management, Principal Sudhakar and all the students and staff for giving this opportunity to speak to you all. So a few words what Dr. Venkatesarulu told. So I will also like to share a few things regarding the industrial academic collaboration, because I was also part of that DSAR initiation, where I was also working with the DSAR and the government regarding this program. This program actually, it is called as Catapult. It is there in the UK actually. So what happens there, as he rightly said, even Novartis companies like MNC, everything. See, the basic discovery research they never do in themselves. Actually, they collaborate with the Cambridge University, Oxford and everything, where the scientists, dedicated scientists are involved in the real research. As he rightly said, because we are pressurized on the work and the atmosphere is so thing is, there are a lot of administrative challenges in the industry where we have to give frequent updates. What happens is, whatever daily we have to give updates, weekly updates and everything. So we get ourselves lost in making the PPTs and everything on the paper rather than really involving in the research and everything. So he pointed out very well what are the challenges and everything. I fully agree with you, sir. But only thing is, I am hopeful that a time will come when who are the scientists who are going to US and UK, they will come and stay in India and do the research. So the humble request is, we have everything in India. It's not necessary we try to find something abroad. The humble request to all the students is, we can only make ourselves proud if we stay here and demonstrate. That means we are a pharmacist. Unfortunately, pharmacist is one field where still it is yet to get a recognition in India. If you see, there are more medical colleges, they are putting nurses, everything. But as a pharmacist, how much we have a recognition in the industry or in the country? It is because we ourselves don't respect ourselves. Because we don't see our profession as a great profession. This is the biggest problem. I see the people who are doing the B.Pharm everything, they just move into the software and everything. They think they want to have a desk job and everything. So today, tomorrow will become very old. If we don't invent the medicine, what are the new diseases coming? So it is the pharmacist who is going to contribute. So my humble request is, be proud, be a pharmacist and try to respect your profession. With these few words, I would like to go to my presentation. So let us just see the topic I selected is containment. As you know, the word itself says that containment. Why you need a containment? You know that now the drugs have evolved so much, where we are also trying to move advanced towards the more drugs like the gene therapy, also the monoclonal antibiotics, virus, and oncology. You know that cancer drugs and everything. These are all very potent molecules. In the sense is, even a microgram of the drug is able to create so much impact on a body. That means while handling, because in a tablet you have only some 0.25 mg or something. But when you manufacture there, you are exposed to large quantities of drug. That means you are handling nearly for 1 million tablet equivalent or 1 lakh tablet equivalent drugs. That means you are holding a very concentrated drug. You know that what you see in a tablet is a diluted version. That is, usually the drug is 1%, 10% or 5% like that. But when you handle the drug, dispensing you know all well. It is the first step where you are directly handling the pure drug. That means the moment of exposure is very high for you. So it's very important that containment is a very important this one nowadays. At our time, when we started as a career as a pharmacist, see there used to be only beta-lactam, non-beta-lactam. I hope you know what is beta-lactam, non-beta-lactam. So there is separate facilities for beta-lactam, non-beta-lactam. Then the steroid came, separate facility for steroids, cephalosporin separate. So people started understanding the science better. That means there is an interaction, cross-contamination, there is adverse effects, everything. Then subsequently a stage came where you know that unpredictable diseases like cancers, everything came where the drugs have to be found very potent. And you know that science has advanced. Now biologics, life science have come, biomarkers have come. Everywhere what happens is we should have more contained environment so that we get the pure drug out of it, and then subsequently we are able to make into the tablets and capsules. So that's why I chose this topic, because I don't know whether your textbook speaks much about this containment, because it's very important that you should know these basics. Okay. Go to the next one. So one thing is, when you say a potent compound, go to the previous slide please. You can see that when you say a potent compound, the occupational exposure limit is 0.03 to 10 micrograms per meter cube. You can see this is, we will see the other terms, but this is what you mean by potent. Just remember this figure. And as you know how we arrived at it, that means this is the concentration which a person, if you know the shift time in the industry, it is 8 hours. That means we calculated how much time the person is exposed, how much quantity. That means one is this is the maximum quantity, 0.03, 0.10 microgram. It will have an impact on you. So automatically how much time you handle the drug, how much quantity you handle the drug, everything comes into picture. That is where it comes that up to 40 hours. That means in industry norms, you consider 8 hours per day into 5 days. You know the drug accumulation. So whatever slowly when you handle the same product again, some part accumulates inside you and inside you, and then subsequently only it goes to the toxic level. Go to the next. So when you say the materials, so if people use the word of materials, that means you know there are processes like this. How many of you have gone to a floor mill? You see the situation of floor mills. Everywhere dust is there, everywhere powder is there. This situation I have faced when I started my career. At the time we didn't have any comfort or anything. We used to have all the ventilation, everything. So what happens at that time, if the compound is very potent, if you are handling, do you think you are safe? You are not safe. So obviously when you are aware, see what I mean to say is, earlier the data is not, enough data is not available. But today there is enough data available so that we can plan how to handle the things. You know the hospitals, earlier it used to be the government hospital, general hospitals, then become the multi-specialty hospitals, super specialty hospitals. So what happens is, we are trying to segregate ourselves so that there is a specialty. You can go to one place where you get everything. And also it is one thing is what you have to do, it is not only, see, as you know the drug harms you, similarly you can also harm the drug. And it is the necessity that there are 3-4 parameters which you have to consider when you are planning. It is only thing is people, environment and your health. So these are the things which are very critical. Next. So when you are handling these high potent drugs, as we know, we have to do this is the exposure limits. What you do is OEL limits. So and we have to consider the OEL limits. Sorry. Also the safety related properties. See, you have seen, how many of you seen aerosol? Have you heard of aerosol? Colloidal silicon dioxide? You have seen how fluffy it is? It is why it is very fluffy, because the particle size is very high. So what if it will float in the air? So what happens, it can go, it is very fine, it float in the air, it goes into your nostrils, everything, it's very irritating. Similarly, that's what you meant safety related properties. So for every drug, there is one called the MSDS. What MSDS means? Material Safety Data Sheet. But when your drug is in the discovery, you may not have the complete properties. But you know the preclinical and clinical studies, during that time, preclinical studies, you get some information on the toxicology and other things. At least it gives what is the dose you can handle at a time. All this information will be available. So the OEL limits and the MSDS becomes a critical for any scientist who starts at the beginning. That means when he is handling the new chemical entity, that means it is a drug which there is not enough data available to completely know the details about the drug. So there are two terms which you have to understand. One is the potency and the toxicity. So the potency, when we say potency, it talks about the therapeutic effect. When we say that toxicity, it shows the adverse effect. So we should have a balance between these two. As you can see, a cytotoxic drug to treat cancer may be extremely toxic, but its potency may be less. That means you need more drug to do the activity. But there are other drugs which require small doses, but it may have less toxicity. So these are the things which you have to see. So potency and toxicity, these two things you have to identify. As I told in the preclinical study, you will come to know more idea about the toxicity. Potency to a certain extent, but after the clinical trials only we will be know about the drug. Go to the next. So this is what I am telling is, the toxicology data usually we get from the preclinical studies, and then early clinical phase, as I told, it's only to determine the optimal doses, balancing therapeutic benefit and what patients can tolerate. It's only an indication, it gives a very broad range actually in the preclinical. At least you know what is the two extreme point. It is like a goal post. That means you know this is the goal. But unless you have a bull, then it won't meet the requirement. Similarly, exposure in this constant and intermittent inhalation of the dust in the manufacturing. As I told, exposure means when you knowingly get exposure. But when you are operating, suppose you are operating some multimill or something. Suppose the mesh which you are using breaks down. What happens? There is a spillage. These are unforeseen situations. So in such situation, you get exposed to this drug and everything. So these two things you have to consider. Now it's just to a quick touch up on some of the points of departures, because you see that these are the parameters which usually get in the pharmacokinetics and pharmacodynamics. So NOEL is the no observed effect chemical causes. It has no impact on the body or anything. And NOAL is the lower than the adverse level. That means it doesn't cause any risk. And LOEL is the low effect level, and LOAL is the low adverse effect, and MTD is the maximum tolerated dose. That means once this maximum tolerated dose is done, then what happens? It will have toxic effect. And ED50 is the effective dose, and TD50 is the mean toxic dose, and then LD50 is the median lethal dose. So as you can see, this whatever the impact is there, these are the parameters which we get in the preclinical studies. So this is just an explanation of occupational exposure limit. Why it's called occupational exposure limit? As you know that occupation means the person who is engaged in the activity. When you say occupational, is because of our occupation we are exposed to the drug. That's why it's called occupational exposure limit. And you can see it is a combination of 4 or 5 factors. As you can see, the numerator compressed factor that will increase occupational exposure. That means the threat is more on the factors, POD, body weight, and euphotic composite uncertain factors. Some of these factors, POD and body weight, which has a direct impact on the occupational exposure. And while the denominator, like the uncertainty factors, and then the modifying factor, volume of air breathed, all these things will have a positive effect. So these things are considered when you do the occupational exposure limit calculation. One more thing is in industry, what we do is for any drug, which is a new drug, we don't have enough information. Using the MSDS, we do a WHRA, workplace health risk assessment, we do it. For every drug based on the MSDS and these OEL limits, bands and everything, we try to do the work level, risk assessment. and we do a surrogated test. Surrogated test means we use a placebo. We don't use the drug, but you use lactose or something and handle it for the time which we propose to do the process, as well as we do the duration of time, quantity. So at the end of the day, we collect the samples and test it and ensure how much we are exposed. That gives the maximum time a person can handle that quantity of material. That's how we determine that. So these are the, as I told, the occupational exposure potential. This is another thing, EP, we call it as a exposure potential. So what are the things you need? As I told, factors to be included are the quantity of material handled and the percent active. As I told, when you see a tablet, the percentage is 1%. If you take a drug, it's 100%. So that is what you mean by percentage. And the potential for dust. As I told, if you take the fluffy material like aerosol, it's very fluffy. But if you take sugar, it's very hard. That means dust won't rise. And duration of the dust, that means how much time you can handle. So these are the three parameters which you have to consider. As you can see, this is the table which is already prepared. As you can see, when you go for 3, 4 and everything, then the risk is very high actually. So on the left hand side, quantity handled per active, and then the right hand side you get the task duration and dustiness potential. So as you can see, if it is an uncoated tablets everything, the risk is less, but granular it is more, and when you take the powder, it is very high. So now come to the select control approach. As you can see there, we have the containment bands. So they have dedicated, there are different bands, the range of the drug means, and then they have the exposure potential. Now this is a matrix where we prepare, and then we prepare the strategy. That means 1, 2, 3, 4, 5. So accordingly, the containment process will be determined. So earlier we have seen 5 stages. As you can see, the first stage, first one EP is 1. Then you can do open transfer, and you can do general ventilation. When you say general ventilation, you have seen in the kitchen. In the kitchen, you have the modular kitchen, you have the exhaust system. That is what this is called a general ventilation. That means you can have similar spot extractor or elephant hands and everything. And when it comes to the band 2, it should have a closed transfer. That means the exposure, usually we should have a closed transfer. And then we have a downflow booth. Downflow booth means it's like a, you can see that, I don't know how many of you know it's a downflow booth. Anybody knows what is a downflow booth? Okay, you have microbiology lab? You have microbiology lab? Yes, sir. Where do you do your inoculation? Laminar flow. So it's a laminar flow. It is called a reverse laminar flow, RLAF. Reverse laminar. That means air comes from the top and goes from the bottom. So what happens is, you are not exposed. Whatever comes, it is swept away. Unidirection it is called. And it is a filtered air. It is filtered through HEPA filters, we call it. 0.3 microns. Why 0.3 microns? Because the bacteria size is 0.3. That's why it's 0.3. So that's what we mean by downflow booth. Open transfer with LEV. That means local exhaust ventilation. As I told, it is like your chimney in your kitchen, modular kitchen. So what happens? Any dust comes, immediately it is sucked out. And then next is the third is the downflow with glove port, shield or glove bag. See, glove port shield means it is like a shield. That means there is a barrier between yourself and the downflow booth. That means you are not directly exposed. There is a shield. You see some of the scooters, everything, you have some windshield. So similarly it is a shield. And closed dust tight transfer with LEV. That means you have closed, means automatic transfer, vacuum transfer happens, but still as a precaution we have local exhaust ventilation also. Any small spill it will be immediately taken. It's local is just like a vacuum. And WIP, CIP included in the process. WIP, CIP is wash in place. That means I don't take that equipment somewhere to wash it. We wash it there itself. Clean in place. CIP is the clean in place. That means we clean there itself. So whatever is there, we clean there and the water is collected in a closed container and it is taken for the further treatment before it is disposed. Flexible containment. That means See, there are isolators. People call it as isolator. That means temporarily you can have that a protected HDFE partition, everything we can have, so that after that one we can remove that one. And when come to the 4 glove box with RTPs, that means glove box is, there is no direct contact. It is like a box. So there are gloves are there. I will show you the picture in the end. So what happens is you handle through the gloves. You don't get directly contact into the material or anything. So that's what you mean by glove box. And then closed dust tight system, that is same. Split butterfly valve. The butterfly valve is you can close it or open it. And then WAP, CAP. And 5 is similar. Only thing is there is it's more tight actually. So once that is done, then equipment and technology selection. So what happens is when you are partly to include materials or compound involved, process being used, environmental safety consideration and GMP requirements. So these are the 4 parameters which we look for the selection of the equipment. You have to understand the purpose. You know there are multi-mills. Multi-mill, there are so many mills are there. Multi-mill, ball mill, co-mill. So which one you select? All these things you have to come into picture here. And this should be a risk-based approach. That means whether there is any risk, as I told WHRA, workplace health risk assessment we have to do. So what is the chance of exposure? How much time you have to do? All these things we have to. See, it's not necessary that we take the containment, but there is a personal protective equipment is also defined. That means sometimes you have to have additional equipments to protect yourself also. It's a second barrier. Good. As I told earlier, we have seen that the nature of the material, it can be powder, it is more stringent. When it is granules, better. Like that, you have to see crystalline means it's less. So if it is heavier, it's always easy to handle. But then it becomes very light because it floats in the surface. The particle size is very fine. You can't see it. So if you see the virus and bacteria, you can't see with your naked eyes. So what is there in the atmosphere, you don't know. So all these things come into picture. Similarly, you have to see the liquid, solid, vapor or gas, which you are using. And then nanomaterials, high adhesion, dusty materials, all these things. Because you know the electrostatic charges. What happens when you mill that one, there is an electric charge is charged. So the particle will adhere and they become granular. All these things will come into balance. And all the flammability, combustibility, reactivity and explosivity. Because when you are a flammable material, probably you should have the flame-proof equipments, motors, everything. Otherwise, it will catch fire. So when you come next is the process. So what happens is, it should be indirect involves direct handling, indirect handling or limited or repeated handling and the number of transfer steps. So what happens is, if you see the granulation, let us see the granulation process, wet granulation process. First step is dispensing, where we are directly exposing the powder. Second step is dry mixing, where all the powder is very fluffy, it is open. Once you have the granulation, what happens? It becomes bowl, wet mass. That means nothing is there. See, I don't know how many of you, at home, if something spills, powder or something, people sprinkle water and then take it to the cloth. It is only the common sense. So what I mean to say is, we are doing daily, but we are not thinking scientifically. So what I mean, the same principle is here. We have a spill kit. When something spills, it is the only thing is, we wet it, then we put a cloth, spill kit cloth, and we take it. So that's what I mean by that is what we meant. And minimum number of transfer points to limit operator exposure. That means if there is a continuous manufacturing is there, that means once I load the material, it shifts, then it moves to the dry mixing, then it goes for granulation, then it goes for the drying, then it goes for milling, then it goes for blending, then it can be loaded into the compression machine, tablets comes, then it can be transferred into the coating comes. So what I mean is the automation continuous process manufacturing is more popular because the exposure of people to these potent drugs becomes very less. So this is what I mean by that one. And also the sample requirements, size, quantity, location, continue, any other limits. See, when you do a blending, how do you know that the drug is uniformly distributed in different areas? So you have to do that sampling. People do top, middle, bottom, 3 corners, middle of the corner, everything do. But if you don't give a provision for a potent compound, suppose you are doing a blender, if you don't, you have to open every time blender, it's very difficult. There should be some sampling. You don't know about the water system, purified water system, what happens is, we give a loop, that means it's supplied to everywhere. But there are points where it is given valves to collect the sample for testing. Otherwise I have to stop, open everywhere. It doesn't happen. You are opening like that. That's what you mean by consider. This also should be considered. How much time we are taking, how much time it's exposed, all these things come into picture. Similarly, maintenance, changeovers, breakdown, all these things. See, what happens is, how the isolators or how the portal companies, whatever is product where it comes contact into the product, means that part will be open, rest of them will be closed. That means what happens is that area will be closed. So what happens is, automatically there is no contamination of drug to the people. So the non-technical area, we call it the technical area. That means the motors, everything is outside, so it can do the maintenance. These are the things which we have to decide. So these are the things I told about. Some of them I covered in the equipment. So cleaning validation is very important. Suppose you have 10 products. Let us assume a cleaning validation. I have 10 products. Each one solve. You know the BCS classification. There is a solubility is a problem. Class 2, class 3, everything solubility is a problem. Then when you are using a poorly soluble drug, then you go to the next product. You have to do the cleaning. So how do you ensure it do that one? So you have to do a cleaning validation. So in a manufacturing, we have two types of cleaning. Class A, Class B, sometimes say Class C also. Class A means, that means you are manufacturing the products one after the other. That means after one batch, you move to the other batch. You don't do deep cleaning. You ensure that previous batch is removed, you clean on the surface and then go. When it goes to the lower strength to the higher strength, that means 0.25 mg, 5 mg, 1 mg, 2 mg, then you always start with the lower strength. Do you know why? Because you start with a high strength, some product is left. When you make a lower strength, the excess drug can get contaminated with this one. So whatever your potency will be impacted. When you go from lower to higher, the chance of drug impacting will be less. That is type B, they call it. And type C is, obviously, it's a product to product. So in industry, what happens is, I can't do every time different procedures. So they make a matrix. That means they take the worst case scenario. That means the limit is 10 ppm, they say. That means after rinse sample swab, it should be less than 10 ppm. They have to get the drug control. So what happens? They will take the worst case scenario. That means what is the worst soluble drug, that they will develop the process, and irrespective of whatever the product changeover is there, they will use the same product. Okay. Next. As I told, environmental protection. See, environmental protection is very important. That means during the process, we may use pipelines, everything, because solvents you need to use. So you have pipelines. You want to do transfer everything. If there is any leakage, what happens? If you are using some common fumes goes outside everything, we are environmentally polluting, knowingly or unknowingly. When we spoil that soil or it goes into the water bodies, what happens? It is taken by cattle, animals, or it is taken by the human also. So all these things you have to consider, because when you set up an industry, there is a called consent for operations that is given by the pollution control board. There are different categories of drugs, green, orange, red. So the pharma comes in mostly on the red category. That means it gives hazardous waste. Hazardous means it is harmful for the human as well as the aqua, environment, everything. So there is a process where there is a control how you handle the waste, bio waste or process waste, how you control the process waste, liquid means solvents. So you have to solvents or the process water, everything. You have to pre-treat. There is a specifications of how much it should be the parameters, pH, ammonical nitrate, everything is there. That parameter you have to meet before you send it to the pollution control for the final disposal. So these are, and you can see that there is a lot of smoke coming. You can see, it's a only example. Why I put it, there is smoke. So it is not good actually, because it is polluting. So all these things will, because your chimney height, because every month I have to monitor the DG carb, exhaust, everything I have to monitor how much stack means. Stack is nothing but the soot, other things coming out. So all these things will come into picture. So let us see what is the containment approach. What are the approach we have to do? So first is the procedure for containment. First is it should be efficient and detailed procedure. That means every data, as Sir told, in industry we have a very meticulously do the procedures. The standard operating procedures we call. For every process, everything, we should have a standard operating procedures. If standard operating procedure is not there, which is a GMP, good manufacturing practices, at least we have a guidance note or work procedures are there. As I told, you should have a primary and secondary containment. That means primary, suppose I have downflow booth. Secondary containment is I will wear a personal protective equipment. These are all the things you have to do. And sometimes you have to do the gap analysis. That means you have to frequently check what are the procedures you establish. How it comes is, we validate the procedures. First we develop the procedures, we do it, we validate the procedures. Sometimes every year we revalidate the procedure. Because year and after, what happens is sometimes the what procedure we have defined yesterday, it may not be valid this year also. Told the startup sequence. See, even for mixing, there is a way of doing mixing. Even the product mixing, we follow a sequence of mixing. Suppose you have a heavy material. I am giving another example. Suppose sugar is heavy. I put sugar in the bottom and I put the aerosol on the top, which is very light. When you start, what happens is it will all fly away. Because the light material when you rotate. So usually we put the light material at the bottom and heavy material on the top. So these are the sequence what I mean to say is when the equipment startup sequence. Cleaning procedure. I explained to the cleaning procedure. Proven standard decontamination procedures. So what happens is usually in industry we have a kill tank. We call it a kill tank. That means what are the process water comes. Suppose it's a virus and when we have to kill it. What will we add some phenol? I am giving in a layman's decision. We put some cresol, phenol in the microbiology. What happens to media? You put some cresol, phenol or something, what happens then? You kill the disinfectant. Similarly, here also we have to do a decontamination procedure. That means, how do you ensure that you nullify the impact of that drug or whatever the components are? And last but not least, highly trained and experienced people. How do you know how to do it? You should have a training. So as I told, so what happens is when you want to have selection process, one is the facility design, equipment operation, safety to achieve the best level. So these are the four parameters which you see. One is the design, equipment operation, safety. So these are the four pillars which you have to consider. See, consideration is the room pressure controls. See, when you are handling a dry powders, usually it is negative. That means if I am using 1%, so the corridor is there. Here you are handling the powder. So usually it is negative. That means nothing goes into the corridor. That means whatever is there, because in that area where you are handling the dry powder, there is a containment. That means you have designed the facility to handle that. You are wearing a PPE. But when it goes outside, what happens is people there, they are not prepared for it. So they get impacted. That's why the pressure difference should be very clearly known. For oral solids, it's always the operating processing area is negative to the corridor. But for liquids and injectables, it is positive to the corridor. Because there the bacteria, everything can't come inside. That's why the processing area is always positive. You should know these differences. That's how you design this one. And airlocks. See what happens is, suddenly if the pressure is always we speak of in pascals. Suppose my processing area, core area where I do the processing, if I want 45 pascals, that means minus 45 pascals. I can't achieve directly. I have to come stepwise. You have gone to the IC units and everything. You have seen the IC unit. This is a hospital, you have gone to the IC unit. First you go, when you go to the IC unit, first there is one area where you wear the apron, then you go inside, there is a lock and everything. So what even the zone, we call it as a zone. We zone different zone, D, B, C and everything. That's what you mean by airlock. So airlock is only to prevent the outside air, outside consumption coming into the inside. It is an interlock. That means when you open the door, this door, automatically some air will come inside. When this door is closed, it can't come inside. So that is the when after the outside door is closed, then you can go inside. Misting showers. Misting showers, what happens is, usually what happens when potent compound, usually change your street goes with the primary gowning inside the facility. While you are handling, you have a secondary gowning. So what happens, once you are done, the powder will be sticking to you. If you move into the corridor, it will be contaminated. So there is a mist shower. So there is a fiery spine spray which is spray on you. Whatever powder is there, it stick to your dress. Then you discard the gown and then you come out. So you are working out clean and the contaminated gown is put separately collected in a bin. That's what my mission. Only for trained employees. And one more thing is these are all process and procedures. Unless you are not trained, nobody will be allowed to go inside. Because you are not aware of what is the threat or the impact. If you go inside, it will have an impact on you. That's why you know certain places we don't take the children because they don't know some of the process or something. Similarly here also. Heating, ventilation and air conditioning. That means you know that some of the process you need a controlled temperature and humidity. How do you handle? This is heating, ventilation. High vac we call it as a. This is the air handling unit we call it. So this will help us to maintain the temperature and humidity actually. And also it gives a purified air. That means it has a HEPA filtration, high air particulate air filtration, 0.3 micron. So there is no particulates or there is no bacteria coming into the picture. And based on the needs, we have the isolator, ventilator enclosures. See, even the isolators have the HEPA filters. So there is a test which we have to do every year, whether the HEPA filter is functioning or not. If it is choked or something, it doesn't serve the purpose. And preventive maintenance and proper changeover control. As I told, you should do the regular maintenance of the equipment so that they function properly. So as I told, these are the two parameters, occupational exposure limits and MSDS. These are the two things which we have to consider when we are handling the potent compounds with personal and environmental impact process. And these should be built in the procedures of the manufacturing. So this is the thing which you have to bear in mind. So these are the two important things. So it's just a definition probably. Potent compound means which can cause chemical, physiological or physical harm to humans or environment. This is what. And some of the examples are the oncology, opioids, vaccines, other components, cytotoxic materials, all these things, organic chemicals, all these things can impact. These are all considered as the potent molecules. Go to the next one. So when you are taking the first step for the safety and health parameters, you should know the OEL limits and the permissible exposure limits. So there is a difference, you know that occupational exposure limit will be the maximum. So what will they always have a lesser limit. So even if it fails, at least you sure that you are you are on the safe zone. So this is another thing which they use as the occupational exposure bands. So we have occupational exposure limits, but they put it as a band. That means they give the range of how much range it comes into each band. If you see the band A, it is 10,000 to 1,000. Band B 1,000 to 100. Band C 100 to 10. Band D 10 to 1. Band E 1 to 0.001. Band F 0.001. So usually band C and D, they also call category A, B, C, D, and also it goes. So usually category band D is supposed to be very potent actually. Band C, some companies even band C, also they follow the same for band 4 process. So understanding process, these are some of the questions we have to ask ourselves. What are the weights, volumes, flow quantities of the material products, which we discussed already. Okay. And then how do operator control the equipment? Do they perform intervention? So how often? That means there is a manual intervention. If it is automatic, once they see what happens is, you know that you can operate from outside also. PLC is there, you load the material, they come out, operate from outside. That's what you mean to say. How when are samples taken, process by QC? How is the material subject packed and where is that done? That means distribution of materials. That means once you do the blending, then you are unloading. So all those things come into picture. What happens when the needs to be maintained, how it is prepared for repair. That means if the material, suppose you are running a product and it stops in between, there is a breakdown. How you remove the material, how you clean the equipment before you take it from in. All these things should be well defined. How the material is transferred through the facility, exactly how is waste contained and removed. As I told, transportation, it's an automatic transfer, backward transpiration, or it's a manual. All these things should be defined. So these things will tell you pinpoint the life cycle and physical route of potent compounds in the facility, where it is exposed, where it is handled, and in what. So these questions will give you some picture how to design your facility, equipment, and how to train your people. See, where there is a manual intervention, they should also understand the need for this one. as I told, points to each step of the process. Now coming to the process, forming of the compound. As I told, it can be crystalline. Crystalline means it's heavy. If it is a fine dust, amorphous, all these things will come into picture. And what is the duration you are going to? Short time is 30 minutes. If it is more, the long time is more than 30 minutes. And the scale of process. As I told, 1 kg. For 1 kg of the batch size, probably you have to have 500 grams. But if it is less, you have to have how much quantity of drug you have to handle, all these things. I think this already we discussed. So now equipment selection matrix. So you know the EPM, exposure potential matrix. We have the OEM band. Then we can select the equipment. The risk information combined. So next we will see the risk information. So based on the chart, what you have seen, we can come with the risk. What is the risk in using this band? Based on the band, everything, we know what are the things we have to take care. So these are the, as I told, these are the different strategies. That means level 1, 2, 3, 4, 5. So when you are level 1, it is only we handle with the general ventilation, with the supporting the operator, dispensing and basic operating tools. This is the general ventilation. When you go level 2, local exhaust ventilation, direct air flow away from the operator, dispensing room, mask and gloves. When come to 3, barrier system with dust collection exhaust, dedicated dispensary, gowned operator. So you can see that level 3 onwards, there is a insistence on the PPE. That means there is a personal protective equipment which is also defined. Level floor, closed landing with isolated glove ports, dive pressure room, gowned operator. And level 5 is robotic handling with no human contact, especially docking, isolated things. So as you can see, as we go further, the human intervention is being reduced because human, we make a lot of this, and even if the robot gets contaminated, it will have no impact on the human. So this is the selection matrix. As you can see that, when the band number comes down, you can go up to F. You can see that the equipment selection matrix is 5. That means you should have more stricter conditions. As you can see, level 2, one level containment. That means there is one barrier between operation person and the material of concern for each additional level. Okay, there is one partition. As I told, some glass partition is something there. At level 3, there are 2 design features such as a barrier and exhaust system. That means you have local exhaust. That means if some spillage is there, it will take care. Level 4 is a isolated and design space negative pressure. That means it's a completely closed environment. So as you can say that individually when it's level 1 and 2, maybe in the same facility you can have, but when it comes to level 3 onwards, it's a plant in plant. That means it's like you have a separate facility inside the plant. That means the outer corridor is there, but you have a separate plant. That means it is contained there. So what happens with contamination, decontamination, everything, so you can handle multiple products. So cross-contamination. I do cross-contamination, whether it's potent, non-potent, everything, cross-contamination is a universal problem. So how do you prevent this cross-contamination? One is the dedicated locker room, separate entrance and exit from the existing plant. That means clean corridor, dirty corridor. So what happens is you enter through one way, you exit through the other way. Separate gowning, degowning, because clean clothes, dirty clothes. So separate gowning, degowning procedures. So unit operation one room. That means you do only one operation in a room. That means you do milling, you do only the milling in that room. There is no other activities. Because when other activities is happening, then 2 persons is there. There may be dusty, non-dusty, all these things will come into it. Single dose design. That means it is a, once you enter, all activities are coming. Suppose there is a wet granulation. All 4 activities will be done in one suit. So that is, you should plan the capacity, that means the capacity matching with all the activities. So for a cross-contamination, we have dedicated what you call the waste drains and the CIP are clean out of place. That means the drain will be collected, directly transferred to a, outside there is one more container, or it will be clean in place. And then the HEPA filters, as I told, HEPA filters, high air, particulate air. And you have the high vac system with the negative pressure. Sometimes whatever one AHU fails, we should have a redundant. That means there should be a backup system. So for the process equipment engineering, you can see some of the features like isolation technology using glove box system, isolated systems, so that what you say, closed transfers, and then dust liquid gas exhaust system. You know the fume hoods that will separately come, and we have a wet scrubber on the top, and closed transfer system with double butterfly valves. That means you change the direction. That means the process came on one direction and it can go to the other direction. And we have the inbuilt bulk container. That means the vacuum transfer goes into the container automatically. See, what happens is in industry also, they have vertical. Suppose you have granulation, usually they keep on the top. So what happens is the granulation happens, then the compression is the first floor. So by gravity they push to the first floor, compression happens, then the coating is the ground floor, coating happens, then it goes to packing. So they use in a flow. That's what you mean by single vertical systems. And sometimes they use a single system disposable. That means just like our food plates and everything, they may use a single bag, fluid bed dryer bags, everything, they use it and throw it away. And maintenance, as I told, it should be properly selected. The waste management also, the collection and the neutralization, everything should be properly planned. And container should be properly. Suppose you have acid-based waste, you should be mixed it. All these things should be segregated. So operator protection, as I told, the PPE starts from second, third phase. Either you should have a full PPE or you have the purified air systems. Powered air purifying respirators, that comes in the band 3, 4, 5. So this is just an example of level 1 containment. As you can see, this is a downflow mode. As you can see, from the top air comes and goes down. So if you handle there is no contamination. Level 2 containment, you can see this is like a dispensing booth. That is open to the room. Here you can see it is enclosed. This is the difference. Level 3 containment, as I told, is an isolator. You have a glove box. So he is not directly touching. He is using the gloves to handle the thing. You can see that one below the other. So upside you have the granulation. Like that it's a sequence, it happens. He has the protective equipment, PPE also additionally. This is just an example of how it looks, a facility. You can see the person enters through a airlock, but he goes through a different route, decontamination lock. And the center is the manufacturing area, and you can see that product enters from the other direction. There is no crisscross between the material and main movement. And you can see the pressure difference. That means the potent manufacturing area is negative. You see the first airlock is double plus, that is positive. And next one is the corridor is positive. And the inside one is negative. That means nothing goes outside. Everything flows inside. Thank you.
Thank you so much, sir, for your delightful presentation regarding containment strategy, handling of highly potent components. Now, I would like to introduce our guest of honor, Dr. Chinta Sailu, sir. Dr. Chinta Sailu, sir, is a principal, University College of Technology, Osmania University. He is known for cutting-edge research in the field of pharmacy. He has a work experience around 27 years. He was awarded PhD in Chemical Engineering from Osmania University. Your achievements such as ISCHG NRC Kulud Memorial Award and other awards reflect a wealth of knowledge that our students can benefit from. In addition, under his guidance, 5 students were awarded with PhD and 3 submitted the PhDs. He has 4 research papers, 22 papers, 4 research projects, 5 conferences papers, and in nearly 22 conference attendances, including 4 invited lecturers delivered in his account. I am glad to invite you, sir, to speak few words about the conference.
Good evening. It is very difficult to talk at the end because everybody will be waiting for the conclusion part. So I take this pleasure to address this today concluding section. And after listening to the giants of industry and their experience, and being there with a current exposure from the industry, so it's a good exposure for the students also. So I congratulate this particular college who has taken a good initiative to make sure that how the industry is being operated and how the students are to gear up to the needs of industry. And I was just listening to few topics